Hi, everyone. Good afternoon. Thank you for joining us today. While people are still joining the webinar, I'm just going to run through some quick housekeeping. I'm Charlotte, and on behalf of the British Neuroscience Association, welcome to the special webinar, Postdoc to PI, Building the Foundation for an Independent Career, which we're very pleased to host jointly with Scientifica. Before we start, just a few rules to go over. We'll be using the chat and the Q&A function today. So if you want to say hello, post any comments in the chat, please do so. If you have any specific questions for our speakers today, please make sure you use the Q&A function, and you can see both of these at the bottom of your screen. There'll be a Q&A session once we've heard from both our speakers. So if you have any questions, feel free to post them and they'll be picked up towards the end of the session. Just to also take this opportunity to remind you that the session today is being recorded with the permission of our speakers and will be available to watch on demand on the Scientifica website following today's session. And finally, just a reminder for everyone to please adhere to the B&A Code of Conduct for webinars to engage respectfully online. So that's it from me. Without further ado, it's my pleasure to hand over to Jeff Ingram, who is the Senior Product Specialist at Scientifica.
Thank you so much, Charlotte. It's a pleasure. Thank you very much to the B&A for facilitating this too. And just want to double check that we're presenting the screen as we planned. Fantastic.
It's great.
Good. So, as Charlotte said, my name is Jeff Ingram, Senior Product Specialist here at Scientifica. It's my pleasure to be here for the second instalment of our Postdoc to PI series. This time, it's building a foundation for an independent career. Now, I hope that Scientifica and I can play a role in your journey to independence. So, where might Scientifica come in? Scientifica specialises in providing global research with cutting-edge equipment designed for electrophysiology research. We're the most trusted supplier of patch clamp electrophysiology systems, whether this be upgrading your existing or inherited equipment, or a scenario where we provide absolutely everything you could possibly need. We combine our products and knowledgeable staff to empower you in neuroscience, cardiology, cancer research, and even more areas. In doing this, we're constantly working with you to improve and deliver the solutions that you need. Over the last 10 years, I've personally been working with researchers like you to get the patch clamp and electrophysiology systems you need to generate that essential early data and get your lab up and running. And you will have your own Scientifica, as we call ourselves, dedicated to you wherever you might be in the world. We can help via consultations, the new lab setups, and upgrading your current equipment. We'll visit your lab, space, and collaborate with you to understand your goals fully. Perhaps you want patch, perform optogenetics, calcium or voltage imaging, or all of the above. We offer expert recommendations for unique electrophysiology needs. We'll make your goals possible with our recommendations and assistance. We'll understand the best way to build your system if you're working in brain slice, a particular region there even, or cell culture, iPSCs, cardiomyocytes, or if you're working in vivo too. We also help with personalized support for budgeting, getting costing sorted on time, and making sure it all physically fits into the space that you have and get allocated. There are so many other logistical aspects to starting your lab. We see so many applications that we know what works, what to anticipate, all really helps the process go smoothly and take some of the pressure off of you. So please do book an appointment with us and start the discussion. It's really never too early. In our last webinar, we covered making the transition to an independent researcher. We had a panel of 4 guest speakers who shared some of their insights and advice on how to make that career step successfully. If you missed it, you can find it on our website in our resources area. Now, today, we're going to take a deeper look into how Dr. Sam Booker, Group Leader now at the Centre for Discovery Brain Sciences at the University of Edinburgh, made this happen for himself. I'll skip the usual summary because Sam will describe all of his history as he talks you through his journey and his experience. Again, if you have any questions, please drop them in the chat throughout. We'll have a chance to visit these at the end. Thank you so much for being here, everyone. And Sam, over to you.
Thanks, Jeff. I'm assuming everyone can hear me. Which is great, thank you. So hi, for those of you I've not met before, which is probably many of you, I'm Sam Booker. I'm based at the University of Edinburgh, and I work within the Simons Initiative for the Developing Brain, as well as the Patrick Wild Centre. And I've known Jeff and the colleagues at Scientifica for gosh, more years than I think all of us wish to remember. Yeah, so I'm going to talk to you today about my experience of making that transition between postdoc and PI and the aspects of that transition that I think are important. I want to preface all of this by saying that, like, the key point is that everyone's experience and everyone's transition to PI will be different. Mine should not be the template, but certainly my experience, I hope, can give you guys some important pointers on how to make that important career jump yourself. So, I'm just gonna advance now. Yes, it is. There we go. So, by way of introduction, I did a degree, obviously. I did a degree in pharmacology from the University of Edinburgh. You might notice I'm back at the University of Edinburgh. I liked it that much. My PhD or my degree was focusing in 5-HT signaling using electrophysiology techniques. I then moved to Glasgow. I don't know why this is not advancing. Where is that? There we go. I moved to Glasgow following my degree, where I did a follow-up PhD looking at GABAergic signaling in the brain. I then moved to the first postdoc in Berlin to continue looking at GABAergic function in the brain. I then moved back to Edinburgh. Again, I liked it so much, where I did postdocs looking at synaptic function in neurodevelopmental disorders. Then I was fortunate enough to be awarded an independent fellowship in 2021 to start my own research group. And shockingly, based on this history, I'm looking at inhibitory signaling in neurodevelopmental conditions as well as other diseases of the brain. So, kind of thinking about this back to front, where am I now? And this is a slightly old photo. This is in 2024, so a year ago. This is my team. This is me there, looking a little bit younger, but not that much younger. And my team of, at the time, 4 PhD students and 1 postdoc. And we're really interested in how does slow inhibitory signaling modulate circuit function in the brain in health and disease. And I think for the large part, that's pretty much like the majority of the actual science that I'm going to talk about. I might kind of touch on flavors of what I do throughout to kind of give examples to what we're doing and why and how you can use your interest to form an independent career. But ultimately, this is our team. So how did we get there? Well, there were a number of questions that came up from the last webinar talking about how you could make it from a postdoc to a PI and kind of key thoughts that individuals have had about what might be standing in their way. So, I've highlighted a few of these questions that came up that were outstanding from the last seminar or the last webinar. For example, how do you choose a scientific question? How and when do you apply for grants? What to focus on during your postdoc to maximize your opportunity for independence? How do you manage your own career as a student at the same time? And kind of touching on how you kind of balance work and life. And, that's kind of, I think we're going to try and go through a lot of these points today. And then if there are any questions, I'm more than happy to take them during the discussion afterwards. So you're you. I'm putting you as the researcher right in the middle of your little world. And around your research world are a number of key aspects that we have to consider when we want to think about during, making that transition to independence. And I'm hoping that for most of you, none of these are particularly foreign. So first of all, you're a researcher. You need to perform research. That's why we're here. As you get more senior, you get the opportunity to perform less research, but you look back at that with rose-tinted glass and say, actually, that's the fun bit. You perform research, you then publish papers. That's that's how you prove that you've done that research. It's the main currency of research and independence. You might want to collaborate. Very few papers these days are performed without collaboration. So, you need to think about this. You need to think about how you're going to fund your research and your papers and your collaboration. And then you've got, the dean of teaching coming and knocking on your office door and saying, well, actually, can you take some honors students to supervise, or can you contribute to this lecture series or tutorial? And that's going to pull at you a little bit. And then finally, after all those things are taken into account, you kind of got to think about, well, actually, how am I going to integrate all of these things, which seem to occupy all of my life, with my own life outside of work. And not unreasonable for this to kind of become overwhelming and complex. And certainly for me, I've found at multiple points in my life, including recently, that these strands can all be overwhelming and that you've got to find a way to kind of weather that storm a little bit. So the first thing I want to highlight, and it's one of the questions that came up from the last webinar, is work-life balance. What is it, and is it tasty? And this actually I find troubling in my role as a new group leader, in the sense that I want everybody in my team or working with me to have the best possible life, both at work and outside of work. I think, the old adage of happy people make good researchers is really true. If you're coming into the lab in the morning and you've had a really rubbish weekend and you've been stuck in the lab and you've been counting synaptic puncture and immunolabeled images, you're probably not going to be feeling as great as if you went out kayaking at the weekend. And I will excuse the old GPT misspelling of balance there. But, it's really important to find time to be able to do what you want to do. And it comes down to the idea of, are you working for your life? Or are you living for work? And I think, there are phases in everyone's career. And certainly I'm not going to tell you that there's not hard periods of hard work to get independence, but you have to have a happy and enjoyable life outside of the lab that allows you to take a break when you need a break from the lab to be able to kind of rest, recuperate, and come in refreshed. So how can you do that? How can you maintain that? Particularly with researchers, and I've moved city a few times, and it is hard. So you move to a new city. How can you find ways to get out of the lab and have a life, so to speak? So reach out to the local groups around the university. It's a great way to start. Most universities have PhD and postdoc societies. They're a great way to meet people that are interested in this. Find time for interests and hobbies, and importantly, stick to them. It's really easy to say, actually, I'm going to scrap off going to play hockey this weekend because I've got this really important experiment. But actually, playing hockey, it'll give you the time to actually take away from the lab and come back refreshed and have a new idea potentially. It's a great way to meet like-minded people, which is actually, incidentally, a nice way to kind of meet people who you might want to collaborate with in the future. And importantly, and this is something I can't stress enough, make up the time for those late nights and weekends. Take time to refresh and recharge your batteries so that you're able to come in and write that grant application or do that pilot data that you really want to do. It's sort of weird to put this at the start, I think, maybe in some ways, but actually, without having a strong foundation of a great stable life outside of work, it's very hard to justify all of the work you need to put in to form an independent career. Right. So you've got your work-life balance sorted out. You've got stuff going on that makes you really feeling jazzed to be in the lab when you're in the lab. Okay, research. So one of the key questions that came up was what to focus on during your postdoc period to maximize your opportunity for a subsequent independent position. And this is a really tricky question. I struggle with it a lot, probably at the detriment to my work-life balance at times. But the first thing you got to think about is, well, you're probably a postdoc or a PhD student working on a grant-funded project. So your PI or your group leader is going to want you to work on the aims and goals of that grant. And as a new group leader, with having spent a lot of time getting grant funding, I want the people working with me to focus on that 100% of the time. But it's not realistic for me to expect that. Actually, what you need to be able to do is be able to find time to answer your own research questions and give yourself the time and space to be able to start developing those questions. The conceptual issue. So how do you actually go about doing that? So for me, I was really fortunate in that I chose and had some exceptionally supportive PIs throughout my career. And I think the number one thing to be able to develop those ideas and your own research is to have conversations early, even at the stage of the interview process or application process for a new job, to identify what support is that PI going to give you in developing your own research. No one's going to say, yeah, you can spend half your time doing pilot data experiments and the other half of the time, 30% of the time working on my grant and 20% of the time doing career development. That's not realistic, but you might be able to find time within your career and your mentorship with them to be able to find support for that independence. And part of that is making time for pilot experiments, nobody develops a research portfolio just from day one without doing a couple of pilot experiments, maybe on a Friday afternoon when you've got that last slice left of the day, or that sample that you've been keeping or you were going to throw away. This is a great opportunity to kind of shoehorn in something that really interests you into your research portfolio, and start small. The best projects or the best pilot data I had during my postdoc period were things that were kind of the same as what I was doing already, but where I could use the same drugs or pharmacology or the same solutions to answer a question that was kind of bugging me, that I didn't know the answer to, and the literature didn't have an answer to, but only involved maybe wild type samples that I could address, and I could sort of start to build pilot data. And it's a great way to find internal collaborators, so colleagues who walk along the corridor, you know along the corridor, or you have a coffee with. Talk about these pilot data experiments. Don't be ashamed. This is what we're here for. Talk about it, and say, hey, that person over there, they're really jazzed about this too, and they've got ideas about how we can answer my question and support that data, and they were interested in helping out using their whiz-bang new technique. And that's a great way to start setting up collaborations. And just as an example of 2 studies of that, the first one here was exactly that small experiment. This was me looking at a brain slice under a microscope on a Friday afternoon and going, what the heck are those cells? Do they do what I'm meant to be looking at in other cell types? And as a result of that, I got a MSc student to work alongside me. And from the basis of that, we then published a paper, which I was first author on. Great. Then, building on that, when I was a postdoc in Edinburgh, I then identified a route that we could do the same type of thing in human tissue and address the same question. And for this, my PI was super supportive and said, actually, hey, we didn't have anything to do with this. This is your stuff. And this is what's led me to be able to start to publish my own work as senior author in my own lab. And doing these little experiments, allowed me to start thinking about finding my research niche, which a few of you asked questions about how you develop that niche whilst in somebody's lab. So the lab I was working at the time was exclusively working on rodent brain. And for me, the big unanswered question is, well, how does what we know in the rodent brain compare to the human brain? And for this, I didn't have access to human brain. I'm not a medic. I'm not a surgeon. I don't interact with patients regularly. But I happened to be at a social event that was organized by the department, and I met a couple of colleagues who were like, hey, you should speak to this guy, Paul. He's a consultant neurosurgeon who is able to who's got regular access to patients undergoing resection of brain tumors. He might be able to help you. And then after meeting Paul, I met Claire Durrant, who also had a similar vision of their research. And together we have now established a bioresource at Edinburgh where we can access living human brain tissue to produce electrophysiological recordings from the human cortex. And this didn't stop with us. This has now expanded out. So in 2024, we had a really, really, really small collaboration of Paul's group, my group, and Claire's group, and some nurses. And now we're expanding, and now we're maybe 4 or 5 groups. And actually, since then, we're now 5 or 6 or 7 groups. So this is now becoming a kind of multi-center initiative from literally having a conversation over a beer and a barbecue organized by colleagues. So you've got your idea, you've got your little collaboration. How do you fund this? And this is really what we need to all be thinking about. Unfortunately, there's no simple answer. We need to fund research, we need to have money. For money, we can apply for internal funds, which are decreasingly common, or we have to apply for external funding. So here's a screenshot of my funding application folder from a few months ago. I've circled in green the ones that were successful at the time. And you can see, hopefully, this is not the majority of the grants. And actually, I'm going to highlight here, and hopefully you can see my cursor. Top one here, this was a small sequence funding. This was £4,000 to bring in a mouse line that we needed for our experiment. It was very small, but since then, this mouse line has been used by about 15 different groups across the University of Edinburgh and has become a central resource for lots of research. The second fund here was, again, a couple of £1,000 for support a summer student, another maybe £1,000 for some travel funding to set up human recordings. And then the next one was a collaborative grant. And this is what I want to get across, is that you don't start out by getting a big MRC project grant or new investigator research grant. You get funding by starting small, aim for those local society seed fundings that allow you to spin out your pilot data into something that might be more tractable to a research council later on. Collaborating can actually benefit from the seniority of colleagues that you've got around the department. So you can actually almost piggyback on these bigger grants to build up some track record and experience of applying for big grants. And think strategically. Think about who is to fund your research. There's no point in applying to a dementia charity if your research is largely based on neurodevelopment, unless you have a very clear reason to do so. And research councils are aware of people applying, that are unrelated to their research, and it's just not going to be a good use of your time. And time is what you have, so use that time and use it wisely, but develop a thick skin, because ultimately you're going to get a fair few rejections potentially, and this is not a personal thing. They just, that grant didn't fit with that funding's call at that point in time. You might go back a year later and you might be successful. So, develop a thick skin and carry on. A few of the questions related to how you can engage with teaching and supervision to support your move towards independence. And I think this is a really valuable lesson. In the UK, certainly, most of the research in the UK is performed at universities. As a university member of staff, you're given the privilege to train and teach students of all career levels and backgrounds. So engaging students, whether it's undergraduate project students, summer students, or master students, early in your career, not only probably does your PI a favor by fulfilling some of their quota of students they're meant to train, but it also gives you the experience of running a small research project. And oftentimes there's a little bit of funding associated with these students. So that actually it gives you a couple of £100 to buy the antibody that you were really worried about asking your PI to buy. But now you can buy it from your own little pot of money that might be attached to your PI, but you can say, hey, I brought this in, can I buy this antibody? And very few people are going to say no to that in reality. Then you can think about applying for funding for summer students, and then once you've had a bit of experience of supervising students, you might want to co-supervise a PhD student to get yourself the experience of actually having a slightly longer term interaction with a student, which allows you to maybe build on some of that pilot data you started out a few years earlier. Right, you've got your students, they're working away with you, you've got a little bit of funding, and you need to produce some outcome. And as I mentioned, outcome and our currency is papers, ultimately. So you're going to want to publish. You're going to want to publish off the grants you're working on. You're going to hopefully try and publish some of the work you're devising. How can you do that? Obviously, you want to start off as first author. That's the aim. You want a big first author paper to set your stage. But as you start to move through these career paths, it's totally okay to ask to be associated with more seniority in the paper. You've supervised those students. You've designed the experiments. You've brought in some of that money. That's totally okay. And actually, the only thing a PI can say to you at that situation is no. And what have you lost? You've not lost anything really. And if you've already identified a supportive PI who wants you to help you work towards independence, they should be supportive of this. And they've developed something like a co-senior authorship, and then ultimately you might end up where you're developing a first and corresponding author, where you've performed the work, but you've also designed the work. And then finally, you end up as the last corresponding author, which then signals to research councils that you are ready to run research programs yourself. So publish regularly, request the authorship that relates to your input, and submit articles to support your transition to independence. And don't be afraid to ask. Right, I'm kind of coming to the pointy bit of this is I have a pathological inability to say no. That's maybe why I'm here. I don't know. It's really important to say no, to learn how to say no effectively. The temptation is, as an enthusiastic and curious researcher, you want to say yes to every opportunity that comes along. And you want to be able to say, hey, I did that and that and that and that and that. But ultimately, you've only got 24 hours in a day, and you've already told yourself you need to spend 8 hours of that sleeping and a few hours of that having a personal life outside of the lab. You haven't got 16 hours to say yes to everything. So try and avoid temptation to say yes, yes, yes. But think really strategically what fits for your work. If you've got someone coming and saying to you, hey, can I maybe use that technique that you and your students have been working on to understand the physiology of, I don't know, AMPA receptors in the olfactory bulb? That's totally okay and might fit with your research. Or it might take 6 months of your life and you get a co-authorship for something that you're never going to go back to. But so think about how it's going to help you, but be collegiate and collaborative. The best science is done when we are open and talk to each other about the work we are doing. And the attitude of having small in-cell labs is thankfully starting to kind of open out a bit. And the better we can be as early career and junior group leaders at being collaborative with each other, the better science will be in the future. But, I'm saying try and say no to stuff, but also you have to say yes to stuff as well. And it's really important as junior researchers wanting to embark on a group leader position that you're saying yes to the people that, may have something to say about your career or your applications in the future. I'm not saying only say yes to the heads of department when they come and knocking on your door, but it's good to build up store credit. And if there's someone in your department who is doing a technique that you think is really cool, and they ask you to do something, they might be willing to kind of scratch your back when the time comes that you would like to use that technique. So it's a very good way of building up a positive relationship in the department, but also be cautious, be able to say no to. Hopefully I've covered most of what was asked previously. There are a few key take-homes that I want to cover. The most important thing, and this is the singular most important thing for a group leader, is it's hard. If you don't love what you're doing now and what you're studying and how you're studying it, it's going to be very hard to get out of bed in the morning to justify having an independent career in that. So really love what you're doing. Find lab group and mentors that give you the room for independence and will support that. Find your niche. It can be small to start with. It can be something that's completely bonkers, but then expand it. If you think it's cool, chances are someone else will think it's cool too. Identify potential funders and grant schemes, but start small and build up. And engage research students early and often. And collaborate locally, distantly, whatever you need to support your research in a mutually beneficial way. With that, I could talk for hours on this, but I probably shouldn't. I want to say thank you to everybody for listening. And importantly, I want to say thank you to the members of my team. The current team is listed over here. And actually, these are all of the research students that we've had in the lab just since we've been independent. And if I was to list all the students who've worked with me along the way, I would need several slides. There's somewhere on my lab website is a full list, and our funders. Thank you for listening. I'm going to come back to that, but if there are any questions, I'm happy to answer them.
Thank you so much. Inspiring, and I'm sure everybody's been quite excited to follow on that journey coming up. There were a few questions that came up while we were talking. Some sort of housekeeping in terms of where the recordings are going to be. So yes, I'll use a chance to direct you all to Scientifica's website. Go to our resource center and webinars. The original one is up there now, and this one will be up shortly once we get things uploaded. They're there, so you can go back, reference key points and take homes. And then, in line with the content today, we've had a few sort of questions. One, perhaps your experience when you were working over in Germany, Sam, would come in. Nicholas has asked, as a foreigner in a new country, the path is complicated these days. Even if your research is great, do you have any advice for us?
It is complicated. The best advice I can give you was actually advice that was given to me when I was a kid. Because I moved when I was about 8, I moved to Scotland from Australia, and I knew nobody in Scotland. And the advice that I was given was take a couple of weeks. Take a couple of weeks, be the quiet guy for a little bit, listen to what's going on around you, work out who the people are that are the right people to talk to. It won't take very long. You'll be on institutional mailing lists, and you'll see who the people are who have a good idea of what the current structure is. And from there, identify possibly a couple of the group leaders who may have just completed this path. So when I moved back to Edinburgh, I had no Scooby how to be independent in the UK. And I applied for 2 positions, one with a junior group leader and one with a senior PIs that I ended up working with. And actually, after I got my job, I went to the office of the junior group leader and said, look, have you got any advice for me? Because I really like what I'm doing, and I think I made the right choice, and I'm glad that you found somebody for that position that I applied for. But I would really like to know how you got to where you got to in this current system. And the best advice you can get is from somebody in your center. I can tell you how to do it at Edinburgh. I can, or not tell you how to do it, but I can give you good advice for Edinburgh University. But what is different between Edinburgh might be different from London, what might be different from Manchester, what might be different from Bristol, and or what might be different from Paris or wherever. And the best thing you can do is find the lived experience in the place that you are working at that time. And then beyond your local center, the other way is to contact people like the BNA or other societies who often have information and packages that are available, or at least advice or mentorship schemes for people to be able to contact and get interaction with. That would be my best advice.
Excellent. And along those lines, Jennifer also asks, because you did a postdoc in Germany, from your experience, do you think that those overseas postdocs are sort of needed for progression or particularly advantageous on your route there? Or?
Oh, that's a tough one. Only because I did it, I would say, obviously. No,
yeah,
I don't think so. I think it used to be, certainly when I was a PhD student, the kind of expectation was that you were a PhD student, you went and did a postdoc abroad, got a different perspective, and you came back to another, or you went to another institution to start your own team. I think, the way that the world and the way that society has moved is that, and certainly research is following that, is that there's not the same requirement to have to move to a different country to do research. I would say it certainly is beneficial to experience different research cultures and different research environments. That doesn't have to be in the same or the same lab, or it doesn't have to be in a different country. And certainly I know colleagues who are at a similar career stage to myself and have been very successful, who have maybe moved within the same country to a different department, or even a colleague recently who's moved from one department to another within the same university to get a different perspective and different set of experience and skills to support their own career. And so I think you can move country. It's a great way to see the world, but it's not, I wouldn't have said it's a necessary requirement. The one thing, the final thing I'll add to that is, though, there are lots of grant schemes for traveling positions like the Marie Curie that are opened up if you move. So if that's something you want to do, there are different funding options available. So it's, it is that thing of trying to tailor it to what you want to do and what schemes you can apply for.
Absolutely. And as someone who will spend a morning in one department and then the afternoon in another just across the road, I can attest to quite how fundamentally different places can be while not being geographically separate, can be on the exact same campus. It's, if for whatever reason travel is not an option to you or it's not what you fancy, you can get that experience by jumping not very far, I'm sure. A couple of other questions have come in as well. While we're talking about venturing out in terms of geography, how about venturing outside of your general interests?
That's a good question. I think it's one of those things. You have to find a balance between sorry, I'll kind of follow that up. By interest, do you think we mean your technical interest or your theoretical interests? In the sense of, I don't necessarily think if you have spent 5 years becoming a hotshot wholesale electrophysiologist, it may not be worth your while to then spend a couple of months going and learning rodent behavior if what you want to pursue as a career is, slice or culture-based electrophysiology. However, if what you mean is more working on a project or a collaboration that is out of your direct sphere of interest, I think that's fundamentally extremely important. I think we as researchers can get stuck in a niche of our own research without thinking of either the bigger picture or of alternative approaches. And actually, I was talking to a colleague the other day about this exact issue of, as a neuroscientist, we get, even as a neuroscientist, which is a reasonably broad field anyway, we get somewhat blinkered into only looking at cell biology and electrophysiology of neurons, or astrocytes maybe, if you're working on astrocytes. But actually, there's a whole world of cardiovascular electrophysiology that many brain slice electrophysiologists won't even look at those papers. And actually, I think by being diverse, you give yourself a much greater perspective of the field, and you develop new ideas. And this is what we want to, we want to be thinking about new ways of answering these hard questions about how the brain or how neurons work. If we want to address those, we need to address them by thinking laterally, which you don't get by following a linear path all the time.
Love it. Perhaps this leads on. Odira asks, is it easier to gain success working in specific topics or fields compared to others that might appear on the outside to be more niche? Tracking things that are in vogue, I think, comes into that rather than...
I think it's a tricky one. My real research interest is working on GABA B receptors. I don't know why. I think I had a lecture as an undergraduate that was really inspiring.
Just got in there.
Yeah, really worked its way in. They're not particularly in the zeitgeist, nor have they ever been. Maybe in 1995 they were. So I think you have the problem. The big problem is that, whilst everybody says that big high impact papers in big journals are not what research councils are looking at or what search committees are looking for when they are hiring, the reality is that people still aspire to those, whether that's right or wrong. I, again, could talk for a long time about that. And certainly my experience is working on the research in the zeitgeist tends to have more success at getting into those kind of coffee table journals rather than other research. Whether that's right or wrong, I don't know. But if chasing that big paper is something you're interested in, or that big study in a big high impact journal, then you might want to think about doing a little bit. But this is where collaboration is great. This is where you can say, we published a couple of papers where maybe the subject matter wasn't my direct interest, but I could see that actually from a career development perspective, being involved in that team and with those researchers would be useful to kind of line my nest of papers and my nest of my kind of stable of collaborations. So I said, hey, after 6 months, I'll do a couple of days a week of those experiments, and that will kind of see me through on that. On the other hand, you might want to really just focus all of your energy and time on what you're interested in. But then that might take longer to reach that big paper.
Yeah, absolutely. Some of the things you've touched on already, I think, might be the solution to this next question. Is we talked a little bit here about, do we publish in big coffee table journals or other journals with our work? What about if you're in the position where you are maybe feeling you don't have strong or enough results to publish regularly? How might you go about compensating that published paper number? Again, collaborations, but any other potential way?
Collaboration is a great one. and where you can kind of come in maybe at a early stage and contribute a small piece of data to a bigger project. The other way to get on, be involved is speak to people in your lab, speak to people in the wider lab groups, and find people who need help with analysis or with data collection for a project that's ongoing, or that's kind of is approaching towards the kind of pointy end of things, and get involved in it that way. Certainly, I've not been ashamed of saying I need a paper out, or I benefit from a few papers in the next couple of years to sort of get my track record up in XYZ, and I can I help out in any way? Is there anything I can do for this? And people often want to help. People want to work together. People want help. We are social creatures. So that's one way, I guess.
Fantastic. Brilliant. Good. Looking through the other questions, I think some of them I'll follow up on after the chat. There was one that came through ahead of the talk, and it was really regarding the balance in pursuing a safe project versus something quite high risk and high impact that you might have on your radar.
Yeah, you should have both.
Yeah.
Ultimately, the way I think about it is you're working on a project with your PI, possibly. I would view that, as an early career researcher, before you get independence, treat that as your safety net. Your PI, if they've got a grant, if that's on a grant or from a foundation bequest or whatever, they're going to have to report on it one way or another. That's going to, which ultimately will either be a paper or a report, both of which, you can find a way to cite or get into your CV. Use students or use research interns, paid obviously, as a way to do your exciting high-risk research. Because, for a student coming in and you say, I don't know, you want to test out this new grab sensor in, I don't know, alligator neurons. But no, getting a student in to culture alligator neurons that you've got from some guy in the zoo, and getting a grab sensor might cost you a bit of money, but probably is not out with the realm of what a MSc project costs or would fund. And for a student to optimize those experiments is an exceptionally good use of their time, and will be a very meaningful dissertation for their studies. But it might not work. And they, from their perspective, they can most MSc projects or undergraduate projects, there isn't a requirement for positive data. So actually, as long as they write it up in a sensible way, they will still achieve excellent techniques and excellent learning and training, but also probably a decent, a very good or excellent dissertation as well. But that gives you the opportunity to focus on something really high risk and potentially high reward that you can then spin out if it works.
Fantastic. Brilliant. Unless there are other questions that appear while I say this next bit, I think we'll say thank you so much, Sam, for talking, and everybody for being here. There's a link's going to come up if you'd like to book in one of those lab consultations with Scientifica. Like I say, it's never too early, and we can really tailor what you need for your stage as well. So that said, thank you so much to everybody. We'll end it.